Okay, thank you. Thank you so much for kind introduction. So, hi everyone. So I'm Juntaro Matsuzaki. I came from Keio University, Tokyo, Japan. So it's really great honor for me to be here, and I'd like to thank the organizers to invite me here and giving me this great opportunity. So I'd like to share some tips to collect blood samples for blood EV research, especially focusing on using the clinical applications. And here I am collaborating several companies shown here. Okay, so as we have already learned that the many kinds of cell types and nanoparticles exist in our bloodstream. So to study blood EVs, ultimately we want to purify the blood EVs by separating from the other components. But we know that it's still challenging, even after the removal of cellular component in plasma, not only EVs, but also proteins or lipoproteins exist. So our society have already discussed for this issue for a long decade, and they published several guidelines or a beginner's guide, as we already learned. So, and the point is here. The point is we should provide detailed information on what we did in the research. So the point of my talk is the same. And for this talk, I'd like to mainly focus on the cancer detection using circulating microRNA. So blood EVs are potential or non-invasive biomarkers for early diagnosis and monitoring of various diseases. And they can also be useful for the personalized medicine or precision medicine. And EVs have various components such as the DNA, RNA, proteins, glycan lipids, and so on. So these components and their combination should be a good biomarker for various diseases. So for now, in this talk, as I said, that we focus on the microRNAs for the cancer detection. So now cancer detection methods are very changing. And so far, the cancer screening methods are mainly performed by the image tests, such as endoscopy or X-ray, ultrasound, or CT scans. But many researchers are now trying to develop a blood-based new cancer detection system called MCED. It stands for Multi-Cancer Early Detection Test. This is a type of liquid biopsy, and ideally, using this system, we can check the risk of various types of cancers all at once. And the ideal scenario is that checking the cancer risk using this blood-based system, and followed by the risk-tailored screening can be received. Here. And for now, a leading company of MCED test is the Grail in US. They mainly focus on cell-free DNA methylation status. And indeed, the prediction accuracy for the tissue of origin, cancer tissue of origin, was very good, shown that accuracy was 95%. But take a look at these panels. So in the stage 1 or 2, the sensitivity was not so very high. So blood EVs and their components should be a better biomarker to do the MCED test. So let's move on the important issue, which is better, serum or plasma? So we already know that the plasma should be much similar to the blood EV components, because to obtain the plasma, anticoagulant is required. On the other hand, to obtain serum, coagulation should be induced. And in any case, double centrifugation is encouraged to reduce the contamination. And here is the comparison. So during the coagulation process, the release of platelet EVs are accelerated, and we may lose some fibrin binding EVs during the coagulation. So considering this point, the composition of plasma EV should be much similar to the blood EVs. So when we see the correlation of microRNAs between serum and plasma, overall the correlation was very nice. And the Max and colleagues published in 2018, they nicely showed that the difference of plasma microRNAs and serum microRNAs. So the different point is shown in red. The platelet-related microRNAs like miR-223 and miR-199 was higher in serum, of course, than in plasma. But on the other hand, the red blood cell related microRNAs like miR-451 are higher in plasma than in serum. And in the other paper also shows that the plasma contains high levels of hemoglobin than the serum. So this suggests that the plasma has a risk of hemolysis or release of red blood cell or derived components. So I think this is my opinion, so I'd like to ask your opinions. But so we can choose. So I think that both plasma and serum have troubles. So we should choose the better one based on the downstream strategies or outcomes. So far, no strong evidence of the outcome for the cancer detection, which is the best. So I found this paper. So in this, the authors of this paper collected the plasma and serum from the same individuals and checked the microRNA profiles. Then they compared the diagnostic performance of breast cancer between plasma and serum microRNAs. And as you can see here, the serum microRNA profiles showed the better diagnostic performance. The reason why the difference can be explained by this concept, tumor-educated platelet. So this concept is well studied in the team in Amsterdam. So they showed that the platelet RNA profiles are quite different between cancer patient and non-cancer individuals. So they said that various kinds of cancer can be detected using the platelet RNA profiles, though the early stage detection was not very good, not very perfect so far. So we know the serum microRNA contains also platelet RNA profiles information. So that's why I guess the serum microRNA can detect several types of cancers greater. So in Japan, the largest scale or nationwide serum microRNA project was performed between 2014 and 2019 under the supervision of Dr. Ochiya. And at that time, I was in charge of a team leader of this project. And we successfully accumulated the strong evidence that serum microRNAs can detect various kinds of cancers. So for that moment, we collected biobank samples from National Cancer Center and National Center for Geriatrics and Gerontology. And we used microarray platform because at that moment, the next-gen sequencing analysis was quite expensive. So we collected more than 20,000 of serum samples and comprehensively analyzed the microRNA profiles. And we divided into the training and test set like this. Then using the machine learning algorithms, we made the cancer detection, cancer discrimination algorithm using the training set, and the performance was checked in the test set. As a result, so x-axis showed the test true diagnosis, and the y-axis showed the prediction result. So as you can see here, the prediction of cancer tissue origin was very nice. Overall accuracy was 88%. And whole data of these microRNAs are already published, and the machine learning codes are also published in GitHub. So if you are, I'm so happy if you can interested in this data and help your researches. Feel free to use. And after that, so at that moment, the previous study was performed using the biobank samples. So after that, we also do the prospective study using the freshly collected serum samples. And for this moment, we used next-gen sequence analysis. And as you can see here, the nice discrimination between cancer and non-cancer was validated. So just last week, the other group, actually, one of the author, the Arcury company is our previous collaborators. But they independently conducted with the multi-center prospective study with Kyoto University in Kyoto. They conducted the prospective test to check the detection rate of pancreatic cancer using the serum microRNA sequencing with machine learning. They showed that the pancreatic cancer was nicely detected even in the early stage. Of course, it was better than the CA99. So yeah, I guess the serum microRNAs is still promising, and keep on studying. It's worth keep on studying. I also introduced the other example, the serum MIR371 test. So is it also already utilized in Europe? So this MIR371 is located in chromosome 19, and these microRNA clusters are completely silenced in our mature human body cells. And these microRNAs plays a role just in the development, just during the development, and we can see the expression in the fetus or placenta. But in some germ cell tumor, MIR371 was expressed. So that's why the serum levels of microRNA 371 was a good biomarker for testicular germ cell tumor. with this kind of very good accuracy. So let's discuss about the stability of blood EV microRNAs. So in clinical setting, the delay before the first centrifugation can be happened. So when we see this figure, as compared with immediate processing, 7-hour delay caused increased levels of hemoglobin. This is the data of plasma. And it was worse in the samples on ice than the samples at room temperature. But in any case, immediate processing should be better. So, and after making the aliquots, we have to care about the temperature of samples. So unfortunately, serum or plasma microRNAs are not very stable, especially in the room temperature or 4 degrees Celsius. And if you want to store more than one month, the minus 20 degrees Celsius was not enough. So, of course, we should recommend to store in minus 80 degrees Celsius. And the methods for RNA extraction or EV purification also matters. So as you can see here, the RNA sequence result was different depending on the methodology of RNA extraction. So total reads and mapping percentage were different among them. And especially if you see the EV proteins, we have to be aware that the contamination of albumin, except for the QEV, which is size exclusion chromatography. And we also have many options to quantify RNA levels. And again, unfortunately, the results of these microRNA levels were different depending on the methodology. So the correlation was not very good among the methodology, and the commonly detected microRNAs are quite limited. So the normalization method to check, to compare the RNA levels among samples should be precisely prepared depending on the methodology, RNA quantification methodology. So in conclusion, again, I'd like to express that the importance of providing detailed information of what you did in this each study. And also plasma is closer to the physiological composition of blood than serum. Be aware of lipoprotein contamination and hemolysis. Serum EV analysis often provides better performance for clinical biomarkers. Processing immediately after the blood draw and store at minus 80 degrees Celsius. Results can be different depending on the EV or RNA extraction or quantification methods. Therefore, we really need the new technology to purify blood EVs. So finally, I'd like to advertise our annual meeting in Japan. We call JSEP in Tokyo, which is coming in this October. If you have a chance to come and join to this meeting, we'd like to very welcome. So thanks for listening, and I'd like to take any questions.
We have time for questions. We have actually 5 minutes for questions, so you can have more questions if you like. Yes, please.
Just wait for the microphone.
Thank you, Rihab Nasser from Lebanon. My question is about the biobank that you did in Japan. Was it circulating microRNA or exosomal microRNA?
That's a total microRNA in serum.
So both circulating.
Circulating.
Okay,
So correctly, serum microRNAs. So
did you have any
it can be different
comparison?
from the exact physiological or circulating microRNAs.
Did you have any comparison between serum and plasma within the same samples?
For that moment, we do not have chance to do that. So yeah, unfortunately. But actually, we do not have the proper samples obtained from the same patient, but we have the serum samples and plasma samples. And after that project, we also tried to validate using the biobank plasma samples, but the result was not so very good.
So your conclusion based on which is better, plasma or serum, was based on previous research, not something you've compared?
Yeah, based on our experience and previous research, I think the serum would be better for the cancer detection.
Thank you.
We have some question online here. First question is serum or plasma better for microRNA biomarkers in non-cancerous samples?
So it depends on the aim of the study, I guess. So what is your thought that there's a setting to check the non-cancer patient, the non-cancer samples?
Let's see. And another question is for RNA-seq. Which library did you test?
We used QIAGEN library kit.
That's great. One more question. What method you use for EV isolation from plasma or serum sample?
Yeah, so for now, we try to use various methods. So as shown here today was actually, we do not purify the EVs. These are the result of total microRNAs in serum. So when we try to check the exosomal or EV microRNAs, we usually used ultracentrifugation, but we tried to use several column-based strategies. Now we are trying to see which is the best.
One question on Slidos. As far as I know, a single microRNA regulates expression of a variety of target proteins. In the sense, I wonder if a certain type of microRNA can be regarded as a biomarker of a specific disease.
Yes, that's a very good question. So actually, every microRNAs can be released from various kinds of cells in various settings, various disease conditions. So it's really hard to say the specific disease just by using just one microRNA. So except for the MIR371 I showed, this is very unique oncofetal microRNAs, which is directly connected with the germline tumor one by one. But in the other case, like MIR122, which is very highly expressed in liver hepatocytes, most of the liver disease, including cancer and inflammation, the levels of MIR125 is elevated. So, and the question is, the target is also varied. So yeah, This is related to the
Regarding to inflammatory disease, one more question is which sample is more useful, plasma or serum? Last question.
Yeah, so for now, as for the making clinical application to detect cancers, yeah, we think plasma would be better, but I guess it depends on the purpose, of course. And especially for the basic research of blood EVs, I agree that plasma is better. Thank you so much.